Yeah, Dr. Abdul Jaleel, can you unmute yourself?
Yeah. Can I start?
Yeah, we can see your slide and hear you. Please start.
Yeah, but are you able to see the slides?
Yeah, we can see the
Okay,
slides.
okay,
Please start.
okay. So actually, the topic given to me was metabolomics principles and the prospects for studying disease mechanisms and identifying biomarkers. So you can very well see that there is no heart in my title or heart failure also in my title. Yeah, that is because I don't work in heart failure or any cardiovascular disease. I work in type 2 diabetes. And another thing is that I am a technology person here. I work in mass spectrometry facility. I am the in charge of that. So that is what my expertise is. And actually, my presentation will be much lighter now because our director has explained many things about what we are planning to do in heart failure. So I don't have to mention that, any of that. So I will briefly tell you what is metabolomics and the tools used for that, its principles, methods, and finally, a couple of results we published using the tools of metabolomics. So majority of you might have heard the term metabolomics, at least the term omics. Omics nothing is nothing but refers to large scale analysis of either genome, transcriptome, proteome, or metabolome. As you can see that it comes to the last metabolome. There are actually other omics also, but these are the main omics field. And earlier, when anybody presents this metabolomics, the direction, the projection from genomics to metabolomics was unidirectional. But now scientists knows that many of these things interact each other and influence. So that is why you see a lot of arrows on both directions. So, but I can tell you that metabolome is much closer to the phenotype. Correspondingly, genome study will be genomics, transcriptome study will be transcriptomics, proteome study will be proteomics, and metabolome study will be metabolomics. So, what is metabolomics? Metabolome is the complete collection of small molecule metabolites in a cell, tissue, or uh, organ or organism. So, what is a metabolite? A metabolite is nothing but intermediates and products of metabolism. They are primary and secondary metabolites. Mostly it is less than 1 kilodalton, that means 1000 dalton mass. And examples are all the sugars, amino acids, peptides, pigments, nucleotides, lipids, etc. Actually, there is another omics which is a recent development that is called lipidomics. If I have time, I will mention that as well. So why metabolomics? As I said, metabolome is the functional endpoint of physiology and pathophysiology, and metabolome is the direct mirror of environmental influence. That is, it shows the effect of nutrition or malnutrition, exercise, treatment, etc. So, regarding the tools of metabolomics, there are mainly two tools. One is NMR spectroscopy, and the other one is mass spectrometry. My expertise is in mass spectrometry, so I will be talking only about that. And with respect to the difference between mass spectrometry and NMR spectrometry, there is a lot of difference. But the main advantage of NMR spectrometry, NMR spectroscopy is that you don't need to process your samples or any derivatization or anything is required for NMR. But with respect to mass spectrometry, advantage is that it is highly sensitive and high throughput. So majority of the metabolomic studies are done by mass spectrometry. So mass spectrometry is an instrument that measures the masses of individual molecules that has been converted into ions. That is, molecules that have been electrically charged. The principle is that uh, uh, a charged particles deflect in a magnetic field, and the deflection depending on the mass to charge ratio of that ion. So this is a schematic representation of a mass spectrometer. You need some area, introduce your sample or the compounds where it has to convert into gaseous phase. So you need a heating element in there. Then you need an ionization area where your all biological molecules has to be, or chemical molecules has to be ionized, either negative ions or positive ions. So you need an electron beam or something to give an ionization. Then there is a magnetic field that is called the analyzer, where the molecules or the ions get deflected according to its mass to charge ratio. And finally, it reaches the detector where it detects the mass to charge ratio and measures it. And it gives you in your computer screen a mass spectrum. That the x-axis is mass to charge ratio. And I will give you one more analogy, a simple analogy, how the mass analysis is done. It is nothing but sorting and counting. For example, like a pocket change of mixture of coins, that is mixture of molecules. 50 paisa, 1 rupee, 2 rupee, 5 rupee, 10 rupees, etc. Then that means molecules of different weight and size. Here it is different values. Then sorting change by value or size. Here separation of mass by charge. So the concept of, finally, the concept of visual interpretation. That is that this is the 1 rupee coin, but it has several coins. 8 coins are there. Here in the 10 rupee, only 2 coins are there. There are 5 rupee, only 4 coins are there. Even though these two are 20 rupees, but it don't merge each other. What is more important is the value of the coin. So the reason, finally, the interpretation like a spectrum. The reason why I showed this simple analogy is that a mass spectrometrist is really never bothered about usually does not bother about the height. What is more important is the x-axis, that is the mass. That is where we identify the compounds, they separate the compounds based on the mass. So mass spectrometry is used to determine molecular weight. That is what I have shown you just now. Then molecular formula, elemental composition, and structure. We do all these things, but to explain all that, we need another lecture. And with all these things, we can either identify an unknown compound, that means an untargeted method, or quantify known compounds, that is called a targeted method. But even though in the earlier slide I showed you a one analyzer, but nowadays nobody uses one analyzer, everybody uses a more than one analyzer in a mass spectrometer, and it is called a tandem mass spectrometry. So what we do is that we fragment the compound into different daughter ions or fragments and detect all the fragments and identify the compounds. So this is basically done in untargeted method also, where all the compounds are fragmented and all the fragments are identified. So in an untargeted metabolomics depends on the mass and or fragmentation database of metabolites identification. We need a database search for identifying the compound based on the mass or fragmentation. So for example, here it is acetone, it is the molecular weight is 58. So when it fragments here, you get a 15 or 43 masses, different fragments. This is the parent ion acetone. This is one example I am showing you. So the database, creation of database is extremely difficult in metabolomics, mainly because of the chemical diversity is very high here. You know that genomics, it is easy because all depends on 4 bases. The sequence difference. Here also in the proteomics also, the sequence difference which is deduced only based on 20 amino acids. But for metabolomics, even though the number of compounds are less, each compound has its own property. That is the issue of database. So then another thing is a targeted analysis. where what we do is that there are different kinds of analyzers that is there. It is usually a triple quadrupole mass spectrometer. So in the quadrupole one, only one of the compound is selected, as you can see here, and in the Q2, that particular one will be fragmented, and in the Q3, only one fragment will be selected, and that fragment will be detected. So, but with that, the parent ion fragmentation to daughter ions is called a transition, and many transition, that means for multiple compounds, can be stacked together in one method, that is called multiple reaction monitoring. So there are, these two are completely different, untargeted and targeted, and both complement each other for biomarker discovery. So this is a simple analogy for that. For an untargeted metabolism, like an SDS page where you see all the protein bands, but in a targeted metabolism, you see only you can pick up only one protein, a specific protein which you wanted, but you know which what you wanted. Here it is like a discovery. And there are the main difference is that information is obtained for a large number of metabolites, and no prior knowledge is required for metabolite detection in the untargeted, and in the targeted method, which require a prior knowledge of metabolites or mass, its mass and fragmentation, and limited number of metabolites can be assayed at once, but that limited number means hundreds. So but it has improved sensitivity over the other one. So here is the usual workflow in our lab. We use either plasma sample, saliva sample, or any biological fluid or tissue. You can separate metabolites from that, and we use different chromatography, and different ionizations also for complete metabolic coverage. And what you see here is that data acquisition and statistical analysis and final data analysis. All these steps are conducted in our lab. So I will tell you two or three examples. One is we have found that distinct postprandial metabolism is in overweight and diabetes, pre-diabetes people. All these people were having no disease and 18 to 20 years of age. We studied normal control and first degree relatives of type 2 diabetes, and one group of overweight people, but they are not obese, and a positive control which are 2 diabetes. What you can see is that the blue color and red color, that is overweight as well as pre-diabetes. Some of the metabolites which are not changed at all at the level, it did not change at all. Here also, this is lysopesis, this is monoglycerides, and oxocolic acid, that is completely different. This is actually, I'm showing only 2 or 3 examples from a large number of metabolites which are different, which we have identified. The same study, we have found that the metabolites are different for sex. There is a sex difference in the metabolites. For example, glutathione, phytostigmosine, lysopesis, etc. They are considered as a good metabolites, which is related to insulin sensitivity. They are much higher level in women, and they are lower level in men. But uric acid, xanthine, and glycodeoxycholic acid type of metabolites, which are correlated with insulin resistance, they are higher in men. So what it shows is that postprandial antioxidant defenses and oxidation products of purine metabolism are associated with insulin resistance in men. Then there is a downregulation in fatty acid metabolism pathway, and accumulation of fatty acid metabolites were observed in the left ventricular tissue of rats with underwent aortic constriction. This study was conducted by Professor C.C. Karta, and the metabolomics was conducted in my lab. And what you can see, that enrichment of fatty acid in lipid metabolites is seen in heart tissues from aorta constricted rats when compared to sham operated and 3 months old rats. One more example I am showing here. This is metabolic signatures of human aortic valve stenosis. This is a collaborative study with St. Bonafide Hospital, Winnipeg, Canada, where we did untargeted metabolomics, and they performed targeted metabolomics of the aortic valve stenosis. So you can see that there is a different gradation for a lot of metabolites between mild, moderate, and severe stenosis as well as calcification. So what this study shows that this was the first step towards the creation of a metabolic map of calcified human aortic valves. The study highlights an independent association of lyso-PA with CAVS severity, and the study also demonstrates that lyso-PA levels are associated with the faster CAVS progression rate. Actually, I think I took 12 minutes already, isn't it? Hello?
Yeah, you have taken your time.
Yeah. So I will stop here actually. Even though I have few more slides, I will stop here because I wanted to talk about heart failure and future work, but that director has already mentioned that actually.
Thank you, Jaleel, for a very instructive presentation. If there are one or two questions, we can take, I suppose.
Dr. Abdul Jaleel, one problem when we face is the stability of the proteins which we analyze. That is one big problem which we face when we study blood samples.
Yeah.
So, what are the methods to overcome that? Can we do something, or it is the inherent problem of metabolomics?
You are actually talking about proteins, isn't it?
Yeah.
No. Actually, we both actually primarily our lab is a proteomics lab. We can separate from tissues or any biological fluid. We can identify or discover proteins actually from your unknown samples. From unknown proteins can be, if that is the question you are asking, we can do that actually.
No, I think Hadi was alluding to the preservation of samples so that there is no protein degradation.
Yeah, that is what.
Oh no! See, the protein degradation actually it happens, but the main thing what we have to do is to immediately store the samples at least at minus 80 degrees centigrade. If you can flash freeze it at liquid nitrogen, then transfer to minus 80 degrees centigrade. That is the best. But this is the only method we can. Otherwise, there is no other preservative is required. Actually, no other preservative is required. You are talking about heart tissue, isn't it? Or serum?
Yeah, serum, because when we try to analyze some markers and transport from other sources to our biobank, it was not visible.
Okay. But this is the only way I can say that. So the transportation, what we can do is that we have to procure dry ice. Dry ice is actually it is a lower temperature than minus 80. Then bring the sample in that. But what happens is that when from the collection center itself, you need to have something to separate the blood into RBC and plasma.
Yeah, that is the most important.
That is what I do because I have a cohort in Alapura district. So when we do the study there, actually what immediately we separate it there. I have a centrifuge there and bring the sample in cold here at RGCB on the same day within few hours and store it at minus 80.
Thank you,
Jaleel. I think Shashi Kumar Gupta is already online. So
okay, yeah.
to the next
Thank you, sir.